Thank you so much to the Royal Swedish Academy of Sciences and the Nobel Foundation for electing me. It's a great honor and privilege. Thanks too to the speakers who went before me. We didn't actually coordinate our lectures at all, but I think it actually worked out really well. And some things have been said before, other things haven't, but we have very different styles, which is fun. So I am going to speak about the birth and future of multi-scale modeling. I think it's also interesting that Martin had a green laser, Ari had a red laser, and I have a blue laser. So my talk is going to deal with 4 different things: how it began, the birth of computational biology, some new work, future work on multi-scale modeling, and then just finally some general things. So how it began. I think it's really important, if you want to get ahead, is to stand on the shoulders of giants. This is always good advice, and I think I was very lucky. Linus Pauling was an enormous influence. I actually gave the Pauling lecture at Stanford a few times and met Linus in his 90s. Another huge influence on me was Francis Crick. He was one of my mentors at Cambridge when I went there in 1968. He was the person who solved the structure of DNA, but really also combined modeling and experiment, and should have given, together with Pauling, respectability to modeling. It didn't quite happen so easily. Then we have experimentalist John Kendrew, played an enormously important part in my life. I'll say more about that. It's also interesting that in the 60s, the most important journals were not Science and Nature, they were Scientific American, because it was the only journal that allowed you to publish colored pictures. And John Kendrew had a cover picture in Scientific American in 1961, and here is Max Perutz, who was in fact John Kendrew's PhD supervisor. Max wanted to solve the crystallographic phase problem with heavy atoms, and he did. For me, Max is the real hero of structural biology, a really amazing person. Finally, David Phillips, the only one of this group who doesn't have a Nobel Prize. I guess Pauling has 2, so it sort of compensates. David Phillips was again has a cover picture in Scientific American, a remarkable man and a really early supporter of computational biology. In the Scientific American article, he shows proteins folding up, done by cartoons. This was a very, very early influence. Okay, another really important influence on the field was Los Alamos and the Manhattan Project. The Monte Carlo method was actually invented there. And here I think is a rather kindly picture of Edward Teller, the father of the H-bomb, and I guess the third author in the Metropolis Monte Carlo method. And finally, and Martin did mention Anansi Raman, who actually passed away relatively young. He had done simulations of argon, and then together with Frank Stillinger, simulations of water. And in many ways, because proteins have many atoms, they are much more like liquids. At least they were more like liquids early on than other things. Okay. So I've told you a little bit of how it all began. So this is basically the scene as it was in the mid-late 60s. So now a little bit about the birth of computational structural biology, which is sort of the field that was recognized and is now an okay field to talk about. So John Kendrew, who had won the Nobel Prize in 1962, had an enormous influence over me. He made a television program in 1964. I had come from South Africa at the end of 1963. I was 16 years old, and I'd never seen television. In South Africa at that time, there was no television. And my uncle and aunt with whom I stayed had a early, I call it black, not black and white, but black and yellow television. And on that, I actually watched 2 things that I remember very clearly. One was the Winter Olympics of 1964, and the other was John Kendrew's program called The Thread of Life, an introduction to molecular biology. And if one just looks at the titles of the various topics he talked about, it could be a current course in molecular biology. I tried to get a copy of the lectures, and I don't think they exist anymore. which is really a pity, because this was one of the first lectures, but it was very influential on me. John Kendrew then actually sent me to Israel to work with Aryeh and Shneur Lifson. And the reason he sent me to Israel was I wanted to start a PhD in Cambridge, and he said there was no space, and after some negotiation, he said, okay, if you go to Israel for a year, then we'll take you. So he sent me to Israel in 1967 to work with this man, Shneur Lifson. This is how Shneur looked back in 67 and sort of how I looked at about that time. This is actually a bit later. I looked significantly younger at that time. Shneur really was the person who got all of this started. He was, if you were to delete one person, if you deleted Shneur, my guess is this field would have been delayed very substantially. And he did this essentially by having a philosophical idea. So again, just to recap, at this time, the mid-60s, we knew that biology is all detailed interactions. Aryeh showed a clock as the insides. For me, what was remarkable in seeing these papers in Scientific American was that the insides of cells were not like a bunch of blood or soup. They were actually like clockwork. Molecules have shapes, and they function because these shapes move. And that's exactly like a watch or a motor car, or almost anything around us. So in some senses, a really good model for a cell would be a city. New York is a great model for a cell. It's not, a bucket of blood. And this just shows the myoglobin structure, where this heme group binds oxygen, the lysozyme structure that was very influential afterwards, where there's a hexasaccharide here and is bound and cleaved. Okay. So basically, it all started with this philosophical idea of the consistent force field, and the idea that Lifson had was that a carbon is a carbon is a carbon. And this might seem completely trivial and obvious, but at that time, people who were doing analysis of small molecule spectroscopy said that the carbon in carbon dioxide was different from the carbon in methane, and so on. There were many kinds of carbons. Lifson said that the force field, or the potential energy surface for a molecule, needed to be consistent. And the good thing about consistency meant that there wouldn't be too many parameters. So in this paper, Aryeh and Shneur basically started to parameterize the force field essentially for small molecules. The force field sort of looks like this. You will notice that I actually have a font which is my handwriting. But when I started using PowerPoint, I didn't have that, and I would actually draw things on paper and scan them in. So this is actually my handwriting. So this just shows what the force field looks like. This force field is really interesting because the key thing was that there are many parameters. So for example, if you look at a bond length, if you take a bond and you stretch it, the energy goes up, and you compress it, the energy goes up, just like a spring. The same thing is true of bond angles, torsion angles. They all have these very simple mathematical functions, and in these functions, there are two kinds of things. So for example, if we take, say, the bond length, we have the bond length B. The bond length B depends on where the atoms are. But then there are parameters. The bond length B0 and the force constant. The bond length is the sort of preferred length of the bond that it vibrates about. Where do these parameters come from? And again, Lifson's philosophical concept was that there would be a small set of such parameters that could be derived in a consistent way. This ended up being very important because one could then very easily take what had been derived for small molecules and apply it to any molecule, with obviously realizing that there's danger in doing that, but the calculations could be done. I often call this the dead physicist's energy function. In fact, I did have dates here, but Hooke, Fourier, van der Waals, and Coulomb all lived at least 200 years ago, and essentially were responsible for each of these terms. What for me is amazing, and I guess Martin did say in his talk, how it's remarkable that things as simple as they were are still being used today, but in many ways, this energy function is still being used, say, by the D.E. Shaw calculations that are 10 million times or 100 million times faster. Okay, so Martin also mentioned the importance of the energy surface, or the potential energy function. This is just an example to show how all the methods, whether they're energy minimization, shown here is just dropping down to the nearest minimum, here shown in 2 dimensions, or normal mode dynamics, which I guess goes back to Galileo, where you vibrate about a minimum, and again, you can do that in 2 dimensions. All molecular dynamics, which was used and certainly pioneered on proteins by McCammon, Gellin, and Carplus, basically involves moving downhill. I'm going to see that now, again in 2 dimensions. So you sort of move along, but you have kinetic energy so you can go over barriers. And finally, the most interesting method, because it came along so late, is the Monte Carlo method. And that basically came with Los Alamos. And again, there should be dates here. I guess I messed up my font, and those dates are not there. But this one, Metropolis, is 19, I guess, well, it was published in 55. It was probably done in like 44. But, you know, Euclid is 325 BC, I believe, and the others are in between. Okay. So the physics behind this is kind of old. The difficult thing was to get it all to work. Now, just to say now about some of the real stuff going on, we felt that the key idea here in the multiscale modeling really goes back to a quote that is actually attributed to Einstein: everything should be made as simple as you can, but no simpler. It actually turns out that there's no real evidence that Einstein actually said it. And if you go to a website called Wiki Quote Investigator, it actually says that a newspaper person, Roger Sessions, actually invented this quote and sort of attributed it to Einstein so he would get extra credit for that. So I guess I'm happy to stick with this as being an Einstein quote. But it's also very important because the reason you need to do this is in order to get a calculation to work, it has to be simple enough to do the calculation, but it shouldn't be so simple that it's worthless. And I often use the example of simulation here as the weather. If you had a really detailed model of, say, Sweden, you could probably calculate the weather tomorrow in 10 years' time. But that wouldn't really be very useful. In order to calculate the model, the weather tomorrow in 2 hours, you need to simplify your model enormously. It actually doesn't really work very well for Sweden. I've noticed that the weather forecasts for Sweden are really, really bad. They're really good in California. In California, they go onto the radio and they say, the weather tomorrow will be like today, and so on for the foreseeable future. Okay, so very much what the work we did was about simplifying representations, minimization with all the non-hydrogen atoms in 1969, atom groups in 75, all atoms and electrons in 76, and then all atoms in water, as Martin mentioned, in 1988. So in many ways, my work has been about multi-scale work. The paper that Arya mentioned, again with Schnell, was I guess my first paper ever published, involved an energy function that was applied to proteins. It's interesting how I actually got the coordinates of the proteins. In those days, there was no protein data bank, but David Phillips had sent Natan Sharon, a biochemist in Israel, the coordinates of lysozyme and myoglobin. And the idea was that someone would actually build a physical model. And I volunteered to build a physical model together with Yuval Eshdat, and actually got my hands on the coordinates and then had them typed up and put into the computer. So there was sort of an interesting connection there. The next paper involved coarse-grained models. And this again, this one was with Arya. And basically, we were folding a protein in a very simplified way. We could make the protein fold up. Now, it became more compact. It didn't necessarily become the right answer. And we did this by combining minimization with normal mode thermalization, which actually meant that you got into a minimum, and then you made the thing vibrate around the minimum analytically and jump out of the minimum. So this was an approach to doing that. Okay. So then Arya, working with me, made this great move into enzyme catalysis, and it's a cool picture of Arya. I had to show that. And again, now you're considering the atoms and the electrons. This is the first movie of protein dynamics that was actually made for me by Richard Feldman at the NIH at an incredible expense. Each frame of the movie required 2 hours of the biggest computer that existed in the USA at that time. And remember, you're seeing 24 frames a second. This movie was actually also very highly publicized by Richard. It's BPTI, the molecule that McCammon, Gellin, and Carplus did the first calculations on. And there are actually here 4 water molecules. And the movie is sort of fun because one of the water molecules starts to move out into space. And it sort of looks like it moves around a bit like a little spaceship moving around here. In previous talks, I've made a reference to David Bowie's Major Tom, but most people don't get that anymore, I guess, except for the viral YouTube movie made in space by the Canadian astronaut. Okay, so moving ahead, it was obviously important to do protein dynamics in water. And this was actually very difficult to do. It basically took us 11 years after the first calculations by the Carlo group, because people had done simulations of water. Together with Ruth Shawana in the audience, we worked very, very hard to try to match the energy functions used for the protein to those used for water. One had to conserve energy, one had to truncate interaction smoothly, but eventually we got it, and here's a picture of Ruth. And the important thing was, as Martin has said, that the movement, the protein moves in water, but basically the movement in water, not only was it slower, but the deviation from the starting structure was a lot smaller. Here it's 1 angstrom, a lot smaller when done in water than when done in vacuum. And these letters just show other simulations done in vacuum, all showing quite large deviations. Okay. A little bit later, together with Valerie Daggett, we actually did unfolding of the alpha helix. And this movie, shown in the next slide, was interesting because I actually made this movie to show to Linus Pauling when I gave the Pauling lecture in 1990. And all the alpha helices have the wrong hand because I made the film backwards. In other words, the calculation was right, but the negatives had been filmed and turned over. Actually, this happened at the Nobel Foundation when we were introduced just the other day, so it's very easy to make. And this just shows an alpha helix in water. The water is there for the calculation, but it makes it really hard to see what's going on. There are hydrogen bonds making and breaking, and so on. We're now going to see the structure at high temperature. So this is at high temperature, 473 Kelvin, which is 100 degrees centigrade, and the molecule very quickly unfolds, as it should. Just the same thing again. I think the final sequence is just showing this as a space-filling model, which sort of looks a bit like a python moving around. Remember, there is water everywhere. We're just taking it out for the movie. This is an old movie made in 1990. This just shows the same thing in space-filling. It's actually fun to write the program to do this, because computers weren't that fast back in 1990. Okay. So that sort of ends the birth period up to about, say, well, probably there's several periods, maybe up to 1988. I now want to say something about the future, because I think that getting a Nobel Prize has a real drawback, so it kind of makes you feel this is the best thing you're ever going to do in your life. And that's a really depressing thought. I mean, that's a great thought to have 5 minutes before you die. And maybe I'll fall off the stage or something. But, it's not a good way to live. You need to have things you still want to do. Okay, so I'm going to talk here about multi-scale dynamics, and start off by saying that one of the key things about multi-scale, you can have simpler energy functions, but you can also simplify a system by having fewer degrees of freedom. And if you think about how you'd characterize a person walking, you can probably have a knee angle, bend, and so on, but you don't have to think about all the atoms moving inside. They're all moving there, but if you said about walking, you'd say there's a knee and it bends and the other leg moves forward, etc. So choosing degrees of freedom is really important. And I'm going to start off by describing several sort of recent calculations of work that is in press or about to be published. This is a study of RNA polymerase, the structure that Roger Kornberg got the Nobel Prize for in 2006. Here we want to try to see the molecule working. This is a large structure, and I'm showing this because Daniel De Silva, the PhD student working with Shui Huang, who's an ex-postdoc of mine, made an amazing movie, which I'm going to show in the next slide. So this is a big system. It's 500,000 atoms, including the water. And this is just a great movie. Now, this is not made by a dynamic simulation. The clock, even David Shaw can't make the clock move so fast for a system of this size. It's made by what they call Markov state simulation, where you think about many different states and you jump between the states. And we're going to see the molecule. This is RNA polymerase, and what it needs to do is move a nucleotide from here to there. This is a fairly simple thing. It's to do with building DNA on RNA. which is what RNA polymerase does. What I really like about the movie is just, I didn't make it, but I guess like Aria's movie for retinal, it sort of has the reaction diagram in the corner. And this is really nice because if you can move the slider backwards and forwards, you can locate exactly when things are going to happen. So now this is going to move across and suddenly move into there. So this kind of shows the reaction, and this is actually fitting quite well what is seen experimentally. I certainly agree with my two previous speakers in that experiment is extremely important. Okay, another thing is involving normal modes. It's actually interesting, I was speaking to one of the winners in physics, and he was saying he was doing normal modes based on group theory. So normal modes are a very interesting idea. They go back, I would say, many, many hundreds of years. And we've just succeeded now in doing normal modes of the entire ribosome with 170,000 atoms. This is a difficult calculation done with Junji Zhang and General Bray, two current postdocs in my lab. They're just about to leave. And this just shows the ribosome. The motion is very exaggerated. These are just the first 6 modes concatenated together, showing them with all the atoms, and exactly the same things calculated with a simple sphere model. And the important thing is that the motion looks very functional. That's all I'm saying at this point. We need to analyze it very carefully. It is very exaggerated, but this is the kind of thing you can do. And this calculation, even with all the atoms, takes 10 hours on my laptop, so it's not a big calculation. It's a way in which you can get the essence of what's going on without doing a very, very long simulation, something that I feel is actually important. The final thing is a Monte Carlo calculation, in this case of RNA, using what we call natural moves. And I'll explain this a little bit in the next slide. This was an idea that Peter Manari, who's currently, was in my lab, is currently an assistant professor in Oxford, and Adeline Sim, who is a PhD student and is now a postdoc in Singapore, and they did. And the basic idea is as follows. So here is a bit of RNA. And what we're doing here is we're not moving every atom. We're moving whatever we like. You can go in there and say the movement should be bend this helix back 2 turns, or anything you like. When you do moves like that, you break bonds. You can't help breaking bonds. And the idea is that you quickly fix the errors you make in the bonds before accepting or rejecting the step. And this works extremely quickly. This just shows that you can see, in this case, the movement of a base pair. If you only move base pairs, the whole structure just moves like this. But you can also move entire helices, and then you're able to sample much, much wider changes of motion. So again, this is sampling the space by the Monte Carlo method, the method developed at Los Alamos. And you can basically do any kind of hierarchical mixture of the modes. Monte Carlo is very nice because you don't need any derivatives. The calculations can be done in a very easy way, and computers are fast enough to make this possible. Okay. So I just stuck this slide in today because it's really important to try to give an example of what this is actually useful for. And it's very easy to say, well, one day all drugs will be designed using these techniques, and I actually believe that. But when will one day be? 5 years, 10 years, 100 years? But I do have actually one example, which is antibody humanization. There I am, which was actually something that is an interesting story. So it turns out, just to make this important, antibody humanization are the most important anti-cancer drugs right now. The idea is you take a cancer cell, you inject it into a rabbit or mouse, it makes antibodies against that cancer cell, and then you'd like to inject those antibodies back into a person. Trouble is that these are mouse antibodies, and people don't like mouse antibodies injected into them. So what you need to do is humanize the antibody. Now, this is an old idea. The idea first came from Greg Winter and colleagues in Cambridge, and they basically said that the way you humanize an antibody is you just take the hypervariable loops, the loops that bind the substrate, and graft them from the mouse antibody into the human antibody. When I got to Stanford in '87, it was a small company, Protein Design Lab, and they wanted to actually, I became a consultant with that company, so almost like part of my deal of coming, although I think Roger Kornberg wanted me to come to Stanford so I could be a consultant for the company and not the other way around. And at that time, I had done work in the early '80s on how you calculate, how you build models of antibodies. I know Martin had done some work like this as well. And I had a computer program which, given any sequence of antibodies, would build you a so-so model. Now, using these models, Carrie Queen who is the main author here, basically looked at the antibody and said, okay, it's not enough to just swap in the loops that are complementary determining regions. You have to also change some of the amino acids behind that. Now, this might seem like a very obvious thing, but when you apply things, it's really important to stick with the obvious. So what they did is they got this idea, they found out, had rules for which residues you had to change, and they patented it. And it turned out that the winter work, which had come before, had also been patented, but their patent didn't work so well, because the winter patent said, just change the hypervariable loops, whereas in reality you had to say, just change the hypervariable loops and 3 more. And that 3 more ended up being worth a great deal of money. Currently, the antibody therapeutics business is at least $20 billion a year. And Protein Design Lab actually is getting something like $500 million in royalties this year. That's the good news. There are 2 bits of bad news. I have no part of the action. The other bit of bad news is this is a patent that was granted in 1989. That means it expires next year. So, but still, I mean, I think this has a number of things. It shows how basic research can be turned into applied research if you wait 30 years. And it's a very clear example. It's also clear to say that by the time I was asked to help do this, it was boring. We had a program that could do it. I said, run the program yourself. But it was still very important. It was also very important to see how writing the patent clearly was very important. Okay, so one could say that I suffer from lack of focus. I'm going to move ahead quickly now. Our work has been hugely pushed ahead by technology. Computers have become 10,000 times faster, 10,000 times cheaper, 10,000 times higher capacity, and 10,000 times smaller. If this was a car, a new Volvo would cost $3, go 1,000,000 kilometers an hour, hold 50,000 people in comfort, and park in a shoebox. So with that kind of tailwind behind you, there's no way that you cannot be succeeding. Family support has been really important. My mother, who's actually 98 and will be coming, yes, often I say 89, but it's 98, and will be coming to the ceremony. She's on her way here. And my wife, Rena, who's in the audience. The old saying, I'm sure you all know the saying, but it's not quite what you think. This was sent by Eric Lindell, who is sitting in the audience, to Rena, and it's a saying that she's been saying almost every single day since then, becoming increasingly surprised. Take chances. It's very important in science to take chances. Here is me backpacking across the Grey Pass in Patagonia last year. I'm actually not doing my email. I'm using it as a GPS to see how much further I have to go. It was really hard. Take chances, but don't be stupid. Again, 2 years ago, I went to Utto, rented a kayak, had never been in a sea kayak before, had looked at YouTube for pictures of how people sea kayak and felt that I knew how to do it, rented a sea kayak in Utto, kayaked about 30 kilometers up to Orno and back again, found a small island over here that I call Paradise. Very beautiful. This is my island, which is exquisite, my tent on the island where I slept, but also really, really stupid because the water was so cold, and this was before midsummers, and if I'd capsized, I certainly wouldn't be here now. So I think it's really important to give advice to young people, be passionate, be persistent, be original, and also be kind and good. You never know when that's going to be important. I just want to finally thank my towering heroes of science, as I call them, Shneur Lifson, who was an incredible inspiration, and I just wish he'd been here, but he would be over 100 years old, and I guess didn't make it. Then my mentors in Cambridge, John Kendrew, my PhD supervisor, Bob Diamond, Francis Crick and Aaron Klug, and Max Perutz, whom again for me is the hero of science. I don't have anything like the groups that either Martin or Aria had. I would say that the distinctive thing about me was I am not one of Martin's Carplusians. I'm almost the only one who's not. But otherwise, he really did cover the field really, really well. I did have a number of people from lots of different countries, have been well supported. I also want to thank the Nobel Committee in Chemistry. Now, this is something which is obviously super self-serving, but I'm not doing it because it's self-serving. I'm doing it because they worked very hard to carve out a new field, multi-scale simulations, and coined this as being a respectable chemistry and biochemical approach. So they have done really, really well. The big winner is our field, but here I'm using the double meaning of the word field in this overlay. This is the American football field at Stanford, where I was asked to appear on the 10-yard line, and 50,000 people are screaming, Nobel Prize, Nobel Prize. Believe me, that is a high which is really hard to equal. And again, my thanks to you all.
So at this stage, I would like to invite all the laureates in chemistry onto the stage. If you would come here, please. Here we go. Just say hello to the audience.